It is my pleasure today to talk about both the value and the challenges of open data for conducting translational neuroimaging research. And I really want to thank the organizers of this session for putting together such a wonderful program. Unfortunately, I'm not able to be there in person today, as you can tell from this video, but I hope that my presentation stimulates some good discussion on the benefits of open data nonetheless. Now, before we dive into talking about how open data can help translational research specifically, I think it's worth drawing a rough outline of what the clinical neuroimaging landscape looks like currently. This is roughly what that might look like currently, though obviously there's considerable variation across different healthcare systems. But generally, people come in at a specific point of care, either on referral from some stream of primary care or as an emergency. And sadly, imaging is usually only done in those cases and not really done as part of a regular health checkup. They might subsequently get a brain scan done on further referral, often focusing on a specific region or a specific tissue type or specific symptomatology that can be imaged, which is then generally still qualitatively evaluated by experts based on their extensive experience and their findings, and possibly a prescribed treatment might be recorded or referenced with an individual's electronic health records. Now, optimizing this largely qualitative and labor-intensive workflow faces several challenges, and sadly we won't have time to discuss all of them in detail, but I firmly believe open science probably has a role to play in each and every one of those, so allow me to very briefly summarize them. First of all, to make optimal use of clinical imaging, there is a challenge of data transfer. In most contexts, data really stays within an isolated hospital system, and for good reasons related to GDPR and privacy. But there's a clear challenge to actually using that data across different systems, or even if a patient happens to visit different healthcare systems. Brain scanning acquisition itself, and to some extent even its storage, is still very heterogeneous and heavily dependent on the preference of the neuroradiology team and the scanner technology that's available in a given context. Also, in clinical practice, there's often very limited processing at all of scans, aside from sort of basic image reconstruction that's often done by the scanner itself. And subsequently, images tend to be mainly visually inspected for a subsequent description might be entered into an electronic health record. Now, while we could discuss all of these challenges, there are, for example, some very promising initiatives, I think, in the open science space that tap into these challenges very specifically. So initiatives such as Open Neuro, but also advances in federated learning systems have really opened the doors for reliable GDPR-compliant sharing of data across sites, or at least trying to make use of that data across different contexts and sites. And community standards around bits have really led to a decent harmonization in terms of data structures and data storage, and it's really tied in very nicely with this development of automated and community standard processing pipelines that are no longer really dependent on the interest or the grace of single groups or labs to maintain them. Today, however, I mainly want to talk about ways that open data and tools can help move an element of this quantitative evaluation into a more, or this element of this qualitative evaluation into a slightly more sort of quantitative process. Specifically, the idea of using open data as a reference point to conduct normative modeling to provide quantitative evaluations and standardized reporting around a person's relative brain health. So as a quick recap for those who missed last year's excellent symposium on normative modeling organized by Andre Marquand and colleagues, normative modeling really aims to move away from this classical case control comparison model that's been very prevalent in neuroimaging research since its inception. So the classical case control approach assumes that cases and controls each form a very well sort of stratified group. And this may often be a reasonable assumption, but especially in clinical practice, there are many other possible scenarios. A clinical population may actually be comprised of sort of multiple subgroups, each having sort of distinct pathologies, as you can see in scenario B on this slide. Or disease-related variation may be entirely nested within sort of healthy variation. It's actually impossible to draw a strict boundary between the two. Or a clinical group may actually be very diffuse and heterogeneous, and as a result, things like misdiagnosis, comorbidities, or an aggregation of sort of different pathologies might actually be detected, such as you can see in scenario B. Now, as you can see in the right-hand corner, I'm not going to go into too much technical detail for this talk. There are solutions for sort of dealing with this, or at least for addressing individually specific deviations. And that's basically this idea of using a population benchmark to address that heterogeneity. And actually, when you sort of squint a little bit, you zoom out on this picture, actually what you can tell is that it really looks a lot like another clinical instrument that most people are actually really, really familiar with. And this is the good old-fashioned growth chart. So this is an instrument that's been in use since the late 18th century, and it's really a prime example of how you can use population data, often even open data, to create a normative reference point, basically to anchor everyone that you want to analyze, or everyone that you want to evaluate against what is the sort of normative or typical presentation of this feature in the population. However, it's also immediately obvious that this actually requires a population representative sample that actually covers the whole age range or the whole sort of context in which you might be, in the clinical context in which you might be acquiring data. After all, the brain doesn't really stop changing at a point where most of these traditional charts stop. So they don't stop after, the brain doesn't stop changing after 18 years, for example. And so to achieve something that really approximates population representation is not really doable by any single group and really requires a community effort and hence the sharing of data. And so this is really where the project that I presented last year kind of comes into play. And so after starting actually from some very exciting open data initiatives such as UK Biobank, Human Connectome Project, ABCD, and many others, Nathan Klein Institute data that I think many people work with, the Healthy Brain Network, and all these other very exciting open data sets. What we really try to achieve with the Lifespan Brain Chart Consortium is trying to pull together data from as many different and diverse sources as we could find, by basically reaching out to people that have published on data sets or people that have shown sort of availability of potential data sets. Now, as you can also very clearly see from this image, is we're still very much lacking data from many geographic locations and age group. We're by no means actually like covering the whole range of what we want to ideally cover to have a population representative norms. And so we should very much consider this an ongoing initiatives. It is, however, already providing some interesting, and I would probably myself say rather robust growth chart looking reference points. So I won't go into too much details of these results or of this effort, as it wouldn't fit within the 10-minute talk that we're already almost coming towards the end of. But perhaps my talk from last year is still online, or otherwise you could simply read the paper. For now, I'll just show that this one graph really illustrates how we can get an idea of trends and variation in the population from these data already. What's, however, much more important about this whole project and specific for this session is the lessons that we learned about using open data and data sharing from this project. And so I'm trying to sort of summarize them here a little bit. But I think the first thing that we learned is actually there is a lot of data that is already to some extent openly available, let alone data that might be clinically acquired and could be accessible through a specific sort of healthcare setting or project. I should also say that in general, our experience with this whole project has been that people are generally very, very happy to share simple data. And I think we got close to like a 97% positive response rate of people like willing to share data for purposes of generating these reference charts. And also, just to add to that, I think in most cases where we did not get a positive response, it wasn't due to any sort of unwillingness of individuals to share data. It was basically because their ethics or their institute really didn't allow them or were very constrained in the kinds of data they could really share. There's also a few caveats or ifs and buts to this whole project that we sort of realized, I would say, probably more towards the end of it, although it's been a sort of learning curve throughout the whole process. And that's, first of all, that this obviously requires a lot of community effort. Like there's no way like any single study could have actually pulled together 200,000 or so brain MRI scans or could have collected those. Also, as you can see from the map, the data that we have are still sort of largely restricted to sort of Western, majority white, city-based, higher income demographics, and that's something that we obviously want to change. And that also means actively chasing up sort of other data sources. And lastly, I think one of the sort of key things that we realized is that this requires high performance computing sort of backbone in the sense that we're currently sitting on like close to 2 petabytes of data, I think now. And well over 2 million CPU hours used to sort of process all these images and generate these models. And I think particularly that last point is really a barrier for entry of these tools into clinical practice, as it takes approximately 20,000 hours to compute and evaluate a single phenotype. And rerunning this whole analysis and updating the whole framework anytime there is a new patient in clinic is simply not feasible. And so open tools also have to consider sort of computational efficiency in their uses, especially since most hospital systems operate on not really the latest type of computer hardware. And so that's really something we try to sort of implement towards the end of our project, and really trying to come up with ways that people could access these massive reference data sets without needing to pre-process or reanalyze anything, and basically coming up with novel ways that we can sort of estimate for new data types how they might be aligned with these reference points. And this is something which we can do with relatively computational ease that could even be run off a website. And if you want to know more about that, you can go onto our website and have a sort of play around with some of the data that is there. And I realize I've already reached the end of my 10 minutes, so I won't show you a full video of this slide, but you feel free to go around on our website and have a play around with this.